Before we begin the seminar, be sure to set your YouTube quality indicator to either automatic or 720p. You'll get much more detail in the presentation, and I think you'll find that useful. Good morning, and welcome to Your Brain, a Personal Tour. This is Steve Saltwick, and welcome to Seminar 1. The subtitle of our seminar today is What You See is Not What You Get, Nor How You Act. We'll talk about two topics today. The first is how does the brain see and perceive the world around it? It's completely enclosed by bone. It has no direct sensory organs. So how does it see and perceive the world? That's the first thing we'll do. The second thing we'll do is look at how we move. We move through space with our muscles. How does the brain control that? It turns out it's a very interesting and complex process, and one we need to be aware of before we proceed. So, welcome to the seminar. I hope you enjoy it. We'll begin with talking about what was that image we saw at the opening loop of the seminar. It turns out it's a very important document. This is a papyrus. The papyrus was written by a doctor in ancient Egypt 4,000 years ago. It records his observations and prognosis as he treated what was obviously a very wealthy Egyptian 4 millennia ago. What's important about this particular papyrus, which is called the Edward Smith Surgical Papyrus, is that it contains the following hieroglyphic. This hieroglyphic is translated as brain. This is the first recorded use of the word brain in written language, and it's contained 6 times in this document. I used this to show that the study of the brain has fascinated mankind for over 4,000 years. Much has been learned. Much remains to be learned. Suffice it to say, when this doctor was examining this patient 4,000 years ago, he had only a rudimentary understanding of what this marvelous organ could do or how to repair it. Much has happened in 4,000 years, and let's take a look at what we can do today.
To Roger Frisch, the violin is not just an instrument, it's part of him, an extension of his passion for music, beauty, and excellence. Roger's mastery of the violin has put him on the world stage. Few have reached this level of expertise. But not long ago, Roger noticed something wasn't quite right when he pulled the bow across the strings.
When I would draw my bow, I suddenly had a shake in my bow. Now, for most other professions, this would not be a concern. For being a violinist, where your career depends on the stability of your appendages, this was a great concern.
Roger has what's called essential tremor, a nervous system disorder that causes shaking. Concerned, he went to Mayo Clinic, where he met neurosurgeon Dr. Kendall Lee.
And the amount of tremor he has would be considered pretty mild, but not in the case of a concert violinist.
Dr. Lee and Roger agreed that deep brain stimulation, or DBS, was their best chance at stopping the tremor. It involved placing electrodes into Roger's brain. But there was a challenge to overcome that created a first-ever situation in an operating room. You see, because Roger's tremor was so small, it would be difficult for Dr. Lee and his team to tell if the electrodes they were implanting were in the best possible place to stop the tremor.
And so what we needed a violin in the operating room.
Kevin Bennett heads Mayo Clinic's Division of Engineering. His team worked with Dr. Lee's team to create a violin Roger could play while awake during surgery.
We built and designed a 3-axis accelerometer that we could attach to the bow so that as Mr. Frisch was playing, we could actually see a plot on a computer screen of the movement that really shouldn't be there.
During the procedure, Dr. Lee inserted electrodes into Roger's brain and moved them to the site where the tremor originated. Once there, electrodes fire electrical signals to hopefully stop the tremor. By measuring Roger's movements, the accelerometer told Dr. Lee if the electrodes were in the right spot. There was no guarantee it would work. Thankfully, it did work.
I thought this was a wonderful challenge to see if this technology could help in this sort of a unique situation.
I started playing right away the day I came home from Mayo. I was back playing with my colleagues with the Minnesota Orchestra 3 weeks later. Without the stimulator on, the tremor is quite pronounced.
You can see the difference. With the click of a controller, Roger can turn the electrodes on and off.
And that's as smooth of a bow as I can play right now.
But when it's on, Roger's mastery of the violin is absolutely clear.
I remain very, very thankful every day that I have had the opportunity to have had this surgery. And, I just do not take it for granted.
Back on the world stage to make music, beauty, and achieve excellence.
I first became interested in this marvelous organ of the body in the early 1970s as part of the biopsychology area in the psychology department of UT Austin. Here's a picture of us in 1975 in front of the old Macy's Hall. I was privileged to work in the lab of Dr. Michael Gabriel, and we were fortunate in the pursuit of some exciting brain research which was published in Science in 1975. I left UT in the late 1970s and took a tour through the high-tech industry. It was great fun. But even more fun was returning to UT and reestablishing contact with several of my major professors. Dr. Mike Gabriel, circled here, and Dr. Michael Damian, circled there, have been of great help with their insight and guidance for this seminar. I really want to thank them for this. I also want to thank Dr. Dennis McFadden, shown here, for not only hiring me for my first job at UT Austin, but for thorough and constructive reviews of the early drafts of this seminar. This seminar would not have been possible without his help and encouragement. I also acknowledge that he beat me in the mustache contest of 1975. And if you're wondering where Steve Saltwick is in this picture, well, here I am, with more hair and attitude and a little less weight than the one in front of you today. Now, your brain sits at the top of your body, on top of the nervous system. It's unique in that it's enclosed in bone. It's the only organ of the body fully enclosed in bone. It sits atop a spinal cord where the vertebrae protect long-running nerves running throughout your body. And then nerves exit those vertebrae to innervate muscles, like shown here in the legs, but also internal organs of the body. Sexual organs, your digestive system, the liver, organs such as the heart, lungs, kidneys, your stomach. All of these organs receive nerves that ultimately pass information back to the brain. Sense organs like eyes and your mouth, etc., are also contained in pathways to the brain. So Ensconced in this skull, your brain receives information about the world through these nerves and through sense organs. Let's look at how your brain senses and drives behavior in a bit more detail. Since it's fully enclosed in skull, it needs some help. That help comes from the peripheral nervous system, that system of nerves that's outside the skull and the central nervous system of brain and spinal cord. Of note is that everything your brain senses is carried by incoming sensory nerves called afferents in this diagram on the left. These nerves carry the familiar senses like sight, hearing, taste, etc. to your brain, but also note that internal sensations from muscles, from organs, it's the way you feel an upset stomach, get carried to the brain via this pathway as well. Now on the right, we'll see that your brain causes behavior by efferent neurons, or neurons leading away from the brain. The vast majority of our time in this seminar will be spent on how your brain causes movement. For this, your brain uses the somatic nervous system. This is the nervous system of the body, of the skeletal muscles. We'll look at some dynamics of this system later in this particular seminar. But there's a large set of other behaviors that your brain drives, and this is using an autonomic nervous system, as shown here. This nervous system will be talked about in more detail on the very next slide. Here, I want to emphasize that control of your organs, the heart, the lungs, your GI tract, is done by this network of neurons, and we are usually not conscious of this control. But we feel its effects via the feedback path as shown on the bottom of the slide. And this is important because this is the way we feel emotions, and that we shall soon see. Now, the brain controls all of the organs of your body in two different divisions. The first division, called the sympathetic division, is the flight or fight syndrome you've probably heard about in high school. This division prepares internal organs for either fighting the tiger or running from the bear. So it'll decrease salivation, increase respiration rate, increase your heart rate, get you ready for a significant struggle, whether it's fighting or flighting. And that is really what a substantial part of your brain and nervous system is devoted to. The other division is the parasympathetic division. This is the chill out and enjoy your meal kind of division. It decreases heart rate. It stimulates the digestive process. It gets you in a position to maximize, extraction of the nutrients from food. And it is the opposite, shall we say, of the sympathetic division. Both are active at the same time, so these things are in a dynamic balance and equilibrium. When the bear shows up, all the effort, as directed by your brain, goes over to the sympathetic division. When you're ready to chill out, all the effort goes over to the parasympathetic division. And then if it's somewhat in between, then these two divisions may be somewhat, they could be and will be activated to a certain extent at the same time. So your brain is controlling this equilibrium, which sets the state of your body. Some anatomy is going to be necessary to become familiar with the brain. We'll keep it at a level like this most of the time. Here's a picture of a brain. The individual would be looking to the left. The frontal lobe is in the front of your brain, next to your forehead. The occipital lobe is in the back of your brain, in the back of your skull. And the functions of the brain do get associated with various lobes, but not in a one-for-one area. For example, primary visual processing is in the occipital lobe. Memory, auditory, some linguistic skills are located in the temporal lobe. The parietal lobe is a multisensory association area. It helps us deal with things in 3D space. It helps plan movements. And the frontal lobe is involved with fine motor control, sensory and motor planning, And those kinds of activities, typically called executive function or associated with human behavior, but we'll see other animals can, display this behavior. But the uniquely human skills, in my opinion, of learning how to learn, overriding certain kinds of habit based on one experience with a sensory cue, those kinds of higher order mental functions tend to be located in the frontal lobe. Also shown on this diagram is the cerebellum. The cerebellum contains 50% of the cells, the neurons of your brain. So half your brain is in this little striped area on the lower right of this slide. The cerebellum is involved in fine motor control. When you strum your fingers, when you learn how to play basketball, when you swing a golf club, when you do a variety of movements that require very precise timing between those movements, it's the cerebellum that control those actions. If we were to go into the brain and look at the cells that make up this marvelous organ, we would see billions and billions of specialized cells called neurons. These neurons electrically are excitable and transmit impulses of electricity down these long, skinny parts to affect other neurons. When a neuron gets close to another neuron, there's a little gap, and when that electrical pulse, the little blue thing traveling down these snaky bits, get close to another neuron, there's a gap, and a chemical is released when that electrical pulse hits that gap. So neurons are both electrically excitable as well as chemically excitable. There's two parts of the ways we can control neurons. We'll use that fact in experiments. It also says neurons can become dysfunctional in two ways. There are electrical ways neurons misfire, as well as chemical ways, the way in terms of neurons misfiring. Each of those lead to different pathologies. Now, many people will tell you there are more neurons in your brain than there are stars in the Milky Way. That's not quite true. There's 200 to 400 billion stars in the Milky Way, and there's, by best guess of current data, 86 billion neurons in your brain. Now, that's still a lot of neurons, but it's nowhere near the number of stars in the Milky Way. What is amazing is the connection between neurons. So, neuron-to-neuron connections are called synapses, and in the typical human brain, there's 100 trillion synapses. Those are junctions between two neurons in a typical human brain. That means the human brain is more complex than any machine ever designed, even on the drawing board, and it makes it capable of rich processing all at the same time. Since the neurons in your brain use electrical signals to communicate with one another, we can actually record this in living brains. The purple signals that are going in between neurons here carry electricity, so we can record that and amplify the sound. If we do that, they make unique sounds. So I'd like to play for you something that we might hear in the laboratory as we were recording from the brains of a living animal. That bursts of pulses are the electrical signals recorded from a cell as it is attempting to communicate with another cell. They have many different rhythms. Some are quite fast, some are quite slow. Some are silent and burst like we just heard. Some burst and then stop to communicate information. There's a wide variety, and each of them have its place in the way your brain operates. The brain has a geometry, and to understand where things are in the brain, we need to be able to cut it at different angles. This shows you the three angles we'll talk about during the course of this seminar. The top one, called a mid-sagittal section, is as if you took a knife and you put it, at your nose between your eyes vertically and then sliced through the brain. You'll see that the surface of the brain, called the cortex, actually folds into some deep valleys, and those mountains and valleys, the peaks and valleys you see on the top in the middle, is actual processing brain tissue that has folded into this cleft in the middle of the brain. In the middle, we see a frontal or coronal section. That's like you took the knife and you put it on top of your head, midway between your eyes and the back of your skull, and sliced completely down. This shows us a good example of the inside of the brain. In the top middle of that, you can see the cleft that comes down through the middle of the brain. And then you can see the structures on the right and the left as seen from the middle of the brain. And then finally, sometimes we'll do a transverse section to show where things are in the brain. And this is as if you took a knife and you put it horizontally just above your eyebrows and slice through to the back of your brain. Structures in the brain have a three-dimensional shape, so they go through all three of these sections. And we need these kind of perspectives to truly understand where things are. And I wanted you to see the mechanics of how we produce these kind of cuts to help you understand where in the brain things are happening. Your brain evolved. And what we show on the left is some representative brains across a wide variety of species. The human brain is at the top left. A mouse brain is at the bottom right. You can see a vast array of sizes, of surface appearance. The more crinkly the surface appearance, the more surface area the brain has. Usually that's associated with more function. And since one thing my major professors always had a rule about is that you couldn't take brains out of students as you studied brain function, so you had to go to animals in order to study, various parts of the brain, because when you did that, you had to take the brain out of the subject at the end to find out, exactly where you were studying. For those of you that are big dog and cat lovers, it's in the middle row right here. For the dog people, you can be amused that the dog brain is slightly bigger than the cat brain. I know the dog people will say that's obvious from the start. I know the cat people will say that's a small cat that we have in this figure. I'll leave it to the dog and the cat people to fight that out. In terms of a family tree, brains differ in their location, size, and organization of functional areas. To use an example, if our common ancestors had a red, blue, yellow, and orange functional area, this family tree on the right shows a representative evolution of that in terms of its differences in size, in terms of its placement, in terms of what area of the brain it is, etc. So one thing we do need to be aware of as we study the brain is from which species are we studying, quote, representative brains. And we have to remember that evolution optimizes behavior in brains subject to the ecological niche of these animals. A monkey eats its food very differently than a rat. A monkey is in a tree that, on a branch that could be going up and down. It is pursuing a banana, is grabbing and reaching for a banana that's varying in space and three-dimensional arrangements. A rat in general finds food on the floor. He does pick it up and eat it, but the mechanics of that reaching and grasping are very, very different between those two species, and we'll see that that has a corresponding difference in the brain. What you are seeing is a depiction of the brain from a project called the Glass Brain Project. This is an actual real-time image of electrical impulses in a human brain that's being studied in a laboratory in the East Coast. I want to transition using this image to one of our first major topics. How does the brain, which is fully encased in bone, sense the world? There are no direct sensory receptors in the brain. You could spoon out tablespoons of brain matter and the patient wouldn't feel any pain. They would probably feel uncomfortable, a loss of function in various areas, but that lack of pain receptors means we can operate on the brain, like with the violinist, in real time. If the brain senses the world through sensory apparatus like your eyes, your ears, etc., how does it make sense of that? How does it perceive the world around it? Is what you see what you get? Or is what you see what you don't get? We'll see it's more of the latter than the former. And that gives us one of our first main insights into the way the brain works. Your brain senses the outside world through a series of awesome sense modalities. Many of them we learned about in high school. We experience them vividly throughout the day. These are vision, which is brought about by our eyes, hearing, which is brought about by our ears and auditory pathways, taste, brought about by our tongue, and olfaction, brought about by our nose. These are a rich set of functions that each have associated cortical areas. As we discussed, the visual cortex that processes the first images from your eye is at the back of your head. You'll see the auditory cortex kind of in the middle in green, and then gustatory and olfaction spread in different locations. The thalamus, which is this orange thing in the center of the brain, it's not on the surface, it's in the middle of the brain, is a relay station. It is important, but it's a relay station, kind of like a railroad switching yard, if you will, sometimes, between the actual sense organs and the ultimate place in the brain where that sensory information is first processed. We're often familiar with these senses that I've just talked about. One very, very, very important sense that we often are not aware of, is in the same sense that we're aware of our seeing or hearing, are the somatic senses. These are the senses that are shown in the bottom right of the slide, and they turn out to be very important in the way our brain constructs certain images and certain perspectives, and I want to be sure we highlight them in this seminar. The first somatic sense I want to talk about is called proprioception, and this is a sense of what the muscles of the body are doing. In the top left-hand part of this slide, I show a silhouette of the arm, and that is the bicep muscle that is connected through ligaments to the top of your shoulder and the lower part of your arm. And then a portion of that muscle is expanded in view in the lower right-hand side. Now, that muscle is innervated with some sensors called muscle spindles. So the nerves that are leading out of those muscle spindles, which are shown in the middle of the diagram, give your brain a sense of the state of that muscle. It is not the nerves that make the muscles contract. Those are different nerves and they're not shown on this picture. The muscle spindles give your brain the sense of, did the muscle move? What is the force of the muscle? Etc. And that turns out to be extremely important, not only for our movements in and out of space, but also for our perception of self. Your brain, when it tells your body to move, gets feedback of the state of those muscles, and the interpretation, we'll see this later, is that you are moving that. It is the feedback from these muscle spindles that is believed to be extremely important in creating the sense of self that we typically assign to consciousness. And I find it very, very interesting that such a lowly sense, something within the scope of your muscles, might be so important in the construction of that conscious construct of self. The second somatic sense I want to talk about is touch. There are a wide variety of receptors in your skin. These receptors can sense an amazing range of forces. They can sense stroking of the skin, fluttering, variation in pressure, details of texture, vibration, stretching, and many other submodalities of these senses. Touch gives feedback to the brain about the status of forces, especially when we grasp objects. Touch is the sense that allows us not to smash an egg when we pick it up. And this is something that the robotic engineers are still dealing with. It's one thing to grab an egg, it's another thing to grab it appropriately so that the yolk doesn't smash all over your face. And that feedback loop, which turns out to be extremely important again in our construction of what the body is doing and the sense of that in space, the sense of touch is extremely important in understanding that modality. It's oftentimes something we really don't think that much about, and it's high time we do. But humans are really visual animals. With all deference to Dr. McFadden, hearing is clearly the most regal sense, but the ones we're probably most aware of and the ones we use throughout our daily life is vision. Now here's a transverse cut through the brain, so this is the horizontal cut, as if we put the knife on top of our eyebrows and slice to the back of the head, that shows you the nerve pathways from the eyes in the front, top right, leading to the back of the brain, that's the occipital lobe, very directly. They do crisscross, they do make one synapse, that's that little relay station shown in yellow in the middle, and then they go to the back of your brain where visual processing begins. We'll study how that back part of the brain and the occipital lobe processes visual information in just a minute. But the first and main point I want to show you out of this slide is that the eyes are directly connected to the back of your brain, so there's primacy given to visual input, and the fact that all areas of the eye are not created equal. The central part of the eye is very, very dense with sensors. So in the central part of your eye, you get the most resolution, you can see the most clearly, and that's where you can sense or detect color. The rest of the eye is not nearly as sensitive, isn't sensitive to color, and so where you are focusing your eyes is extremely important to your brain as it constructs the visual world. Using computers, we can map where the central part of our eye, this is called the fovea of your eye, we can map where it is being focused. So in this diagram, the little red dot basically tracks where the eye of the subject is focusing on. Where that red dot is, that's where the center of our eye is focused. And that is where the brain is getting the majority of information about the visual field. Let's play a short film while we track this subject and watch what the eye focuses on. So what did the eye focus on? To me, it was almost exclusively the faces of the two people involved in this interaction. You didn't see the red dot survey the room. You didn't see the red dot look at the clothes in the closet. It almost exclusively focused on the faces in that visual scene. So your brain, when it's interpreting this visual space, has got to build up a picture of two individuals conversing, one has just come through a door, etc., from a scene that's built up over time, and from a scene where some parts of the scene are getting virtually all the visual attention compared to the other one. Now that's an interesting way to build a picture of the world. So after the eye has focused on an object, and the nerve impulses have gone to the back of your brain, how does the first stage of visual processing occur in the brain? Well, it turns out the best way to think about this is that the first level of visual processing is detection of elementary objects in very precise locations at very precise orientations. On the left of this screen, you see a monkey that is in an apparatus where we can study his visual cortex. In front of him is a screen. We can project lighted bars or other objects on this screen, and we study the response of neurons when we present those visual stimuli. I will tell you in this and all other experiments that we were doing with animals, rest assured that this animal has been treated in a humane way. Great care is given to prevent discomfort from the animal. All surgical procedures are done under anesthetic, etc. There are quite strict rules governing animal experimentation today. So even though this may look a little stressful to the animal, and I guess he really would be rather playing in the trees of Africa or someplace, great care and great consideration is given to the animal, and I won't say that on every slide, but this is the first one, I think, that we've presented this way, and I wanted to make sure that was clear. So here is a neuron shown on the right that we say is preferentially tuned to a particular stimulus. This stimulus is a rectangle oriented at about 45 degrees, I would say, from vertical. And when that passes the area of the visual field that this cell is responsive to, it sounds like this. So that's a very robust sound when the stimulus is correctly oriented and placed in the correct part of the visual field. You can try other stimuli in this place of the visual field, so in its receptive area, and if that stimulus is vertical, you get a slightly less response, as shown on the top orange portion of this diagram. Tilt it the other way, and you get no response at all. This is an excellent description of how first-order visual processing occurs. Cells, neurons in your visual cortex are tuned to very precise stimuli in very precise places in very precise orientations. What happens after the brain processes this at a first level of detail? We've seen how it's being analyzed for very simple shapes in very precise places at very precise angles. What happens next? Well, various areas of the brain back in the primary visual cortex or surrounding the visual cortex, all at the same time, analyze the visual field for some basic information. Color. Where are the various areas of color? Motion. Are parts of the visual field moving? And if so, how are they moving in relation to one another? Understanding that those set of dots that seem to be moving behind the little bush next to you could be a saber-toothed tiger, that's a good thing for your brain to be aware of. The form of the image is analyzed. Does it correspond to objects that we might recognize, etc.? That's in a different part of the brain, but the form, the kind of generic form of the object, is going to be analyzed at the level of visual processing early on. And then finally, you have two eyes. We are binocular. We can detect depth of objects. So a 3D image of the object is built up in parts of the brain that give us a sense of, how thick and thin and, what is the position of the object in three-dimensional space. Well, what happens next in the visual cortex? Well, in subsequent areas of visual processing, there are many visual processing and association areas. The next stage is breaking down the visual image into fundamental building blocks. Those fundamental building blocks, examples of those, are given on the left-hand side of this slide. They're called geons. They're canonical objects. You can think of them as Lego blocks of fundamental shapes. And by taking these fundamental shapes and merging them together, the brain is able to build representations of fundamental objects. For example, if you want to build an object of a paint bucket or a sand bucket, a water bucket, you can take the shape of a curved handle, number 5 on the left, and a cylinder, number 3 on the left, and put them together to form an object of a bucket. Likewise, the object 5, a little loop, a handle, now becomes a handle, coupled with a cylinder, number 3, and a slightly different scale, becomes a coffee cup or a drinking cup. There's good evidence that those objects on the left, called geons, are subject to fundamental analysis by your visual cortex and then merge together in various ways to form object representation. So this is another core principle of your brain. These objects are being analyzed all at the same time into canonical forms, and then if a telephone is present, it is seen as the merger of three geons in this instance. And this is the way your brain is building a perception of the visual world by fundamentally merging canonical objects, these geons, together into more and more abstract forms that meet what we see in everyday life. Well, what happens next? Take a look at this diagram. What do you see? If you look at it and think about analyzing it, but as you look at it, there's two ways to see this. One is a set of blue lines and Pac-Man-like circles sitting on a flat plane for no apparent good reason. The other way to look at this object is a solid white triangle suspended above a blue triangle and three circles. I hope once you hear it described that way, if you didn't see it that way before, that is the way you're perceiving it now, because that's the way most humans would describe what this is. It is not an arbitrary collection of line segments and funny-looking circles. It is a white triangle suspended above a blue triangle and three circles. What you have just done is made a perceptual decision. You have applied a schema of space. You have decided these are the way objects are represented in space. You've analyzed the geons present and basically decided they represent forms of triangles. So just like we've talked about in the preceding slides, you've analyzed this for color, you've analyzed this for form, you've analyzed this for 3D space. And the reason this is important is that most research now points to when your brain makes that kind of perceptual decision, that is when these objects really enter into consciousness. We're really not aware, unless we focus and attend to them, we're really not aware of when the brain is doing fundamental analysis of the visual field. The line segments, the geons, etc. That's not conscious in our experience. But when we decide on a perceptual framework, when we have built these geons into objects like triangles, when we have put them in three-dimensional space, that is when these objects enter into consciousness. Now, in everyday life, your brain is presented with a very complex visual field. When you look at ordinary objects in front of you, you see objects that are built up of objects, compound objects, if you will. So, for example, the plate of fruit being shown in this diagram is really a compound object built up of banana slices, a plate, what looks to be whipped cream, some fruit compost, some slices of oranges. So objects are built out of objects. This is way above the level of the fundamental geons and etc. But I think it is a fundamental property of the brain that objects are created from objects. So low-level analysis builds geons. Those geons build certain objects like coffee cups, forks, what have you, plates. Then those are built up to more and more complex objects like a plate of fruit. And in reality, the compound objects that can be built by your brain can become astounding in terms of their complexity.
Those biomarkers and our capacity to measure them leads us naturally to Jennifer Aniston.
if we could have the next video. So we have very rarely the occasion to measure the response of the brain at the level of individual nerve cells, because for obvious reasons you can't access the nerve cells in a human brain. We can only do it by and large in experimental animals. But sometimes, particularly during neurosurgery, you can actually listen to individual neurons. Now inside the brain it's silent, right? There's no sound. But if you put in a microelectrode and you can listen to the individual nerve cells chat to each other, you can hear, in fact, I think we have a recording, they have this sort of staccato sound. If you take the electrical signal and amplify it and put it on a sound speaker, you can actually hear them talk to each other. Each one is a little, each tick is a little action potential, it's a little pulse. So this is actually a recording from a surgical measurement where the neurosurgeon put a microelectrode into the brain of a patient, and this nerve cell responded very selectively only to pictures of Jennifer Aniston.
I think it's fair to say Team Angelina.
It is true. It is true. It was because why Jennifer Aniston? It's not that we're born with Jennifer Aniston neurons, at least hopefully. But we're born with a disposition to learn things So, you know, your mother, your parents, the president, famous actors and actresses. So here, by mere chance, we happen to come upon a neuron that selectively encoded the presence of Jennifer Aniston. And sometimes you find cells that even respond to the name Jennifer Aniston or to the name of the famous person. It could be your friend, could be your mom, could be a famous actress.
I'll note that during surgery, this particular cell responded to pictures of Jennifer Aniston, not just in, full frontal face, but also of side views like you might get on a police plotter, line drawings of Jennifer Aniston, and as the speaker talked about, even the name Jennifer Aniston evoked this particular cell. Similar recordings have been made in different patients with pictures of Homer Simpson and other comic characters. Speaking of Homer Simpson, I'd like to engage him in helping summarize this section of the seminar, and we'll use him throughout the seminar to help elucidate the main point of each section. So here we have an x-ray of Homer Simpson. You can see his brain is slightly smaller than the brains we've been discovering, but that's because his brain can only hold one important point at any one time. And the summary statement I would say for this segment of the seminar is that your brain senses stuff, line segments, geons, whatever, but you perceive objects. So it is this fundamental analysis of sensory perception, what you see, is not necessarily what you get, because you get objects. Those objects can be formed from memories, those objects can be formed from fundamental shapes sometimes. Objects, most importantly, can be formed of other objects, but those objects enter consciousness, and that's when you're really aware of things, when the various sensory modalities have been analyzed to a level of object. And that is the main message of this first half of our first seminar. Now let's turn our attention to the second half of this seminar. It's really the other end of the spectrum, movement. We interact with the world around us by moving muscles. We change the position of our limbs, we locomote in the environment, we turn our heads, we focus our eyes. So in this part of the seminar, we're going to look at how does the brain do that? And more importantly, we're going to see is there something that is equivalent to objects. On the sensory side of the equation, we saw how initial sensory input was analyzed at a very low level and then abstracted into objects. In fact, those objects were extracted into objects of objects. Well, is there something that goes on like that in the movement system? Let's find out. The first hint that there might be something like movement objects was discovered in an experiment from 1977. An enterprising young psychologist asked several subjects to write the same sentence with a variety of appendages. This particular sentence is a palindrome, so it says the same thing whether you spell it from left to right or right to left. This is a particular study point of Dr. Dennis McFadden, but this is not Dennis's handwriting. I can tell. It's much better than Dennis's handwriting. What is clear from looking at this picture is that there is some template, there's some kind of template that represents the cursive writing on this, and then that template is expressed in a variety of muscle movements. And here, between the right hand with the wrist restrained, the left hand, the teeth, the pen was actually held in the teeth, and even at the bottom, the pen was strapped to the right foot, and the subject wrote these sentences. Now, clearly, fine-grained muscle movement does vary across these samples, but I would look at this and say, there is some very interesting evidence that points to a particular movement template that then is expressed by different movements of different muscles, but again, expressing the same object of this overall movement pattern. Now, in the search for movement templates, there's always a controversy. Does a purported template reflect the actual movement itself, or is it really just the muscles in some fashion that were being represented and expressed in any kind of part of the brain that looked like it reflected a template? So, in what I believe is the classic experiment that showed, yes, indeed, there were movement template neurons. This was conducted by a gentleman named Umlita in about 2007, if I recall correctly, and it used two different kinds of pliers. The normal kind of pliers, being shown on the top half of this screen and being held in a chimpanzee hand, is normally open, and then to pick something up or grab a piece of food with it, you would squeeze the pliers in order to hold the object in the tongs of the plier. On the bottom of the figure, you see something called reverse pliers. Now, these function exactly opposite to normal pliers. If you've ever used these, you'll know that it takes a bit of getting used to. To open the tongs, you've got to squeeze, and then to grab the object, you relax the hand, as shown in the bottom of this figure. So the set of muscle movements, the activities of those muscles between the normal and reverse pliers are exactly the opposite. If we could get something that represented grabbing the food or grabbing an object, and it was the same between normal and reverse pliers, then we have definitive evidence of a template for movement. Now, during the experiment, Umlita looked at monkeys using these pliers to obtain bits of food. Here's a video of one of the monkeys using normal pliers. The red lines coming out of his eyes is a computer animation of what the monkey is looking at. Remember, we saw the red dot in the previous part of the seminar showing where the eyes were focusing. This shows you in a similar fashion what the monkey is focusing on and looking at. This is not Super Monkey. These are not lasers out of his eyes or something like that. It's merely showing us what he's looking at. And if we look at what he is doing, he's using normal pliers. So now he's grabbing the food and bringing it to his mouth. Remember, he is squeezing in order to get this done. Now let's take a look at the same monkey using the reverse set of pliers. In this instance, he's looking at the food again, and now I'll let it go a couple of times where you can see he's squeezing to open it and then letting go to actually grab the food. In the same monkey, we found cells that corresponded to the grabbing of the food, the letting the pliers close around that bit of food. The red arrow shows when in time that was happening, and then the neuronal activity is shown in two different fashions above. For unit 210, that's one of the brain neurons we studied, each little tick mark in the top half of the white portion of that record is an activity of a given neuron. There's many rows of trials, so each time the monkey reached for the food, it is a different row along those tick marks. And then we see that there's a neuron, that neuron 210, is very active just before the pliers close around the food. In the bottom half of the white graph, there's a histogram. So it basically counts the number of spikes and graphically shows a peak just before the food is obtained. Likewise, a different unit, same monkey, unit 199, shows another neuron that is focused on, the precise point in time that the monkey is grabbing the food with the normal pliers. Now, that same monkey, those same units were observed when the monkey was using the reverse pliers. Remember, the muscle movements are exactly the opposite. However, the behavior of the units of the brain are exactly the same. These two units, Unit 210 and Unit 199, and they looked at several others, basically encode for get the food. It encodes for the act of grasping that piece of food regardless of what muscle units or what muscle movements are involved. And that is an object for movement. If you stimulate areas of the monkey brain, and the monkey brain here is shown looking to the left, so the frontal lobe is on the left-hand part of this diagram, and the occipital lobe, where our visual processing occurs, is on the right-hand side of that diagram. You will find specific areas where specific behaviors are elicited from stimulating those areas of the brain. These are movement objects. They are composed of other objects, which ultimately result in the movement of different sets of muscles. There's reaching, climbing, leaping, chewing, licking, defensive postures, moving your hand to your mouth. Some people actually prefer the word vocabulary in this. I'll use the word object since we kind of focused on that from the start of our seminar. But there is a rich repertoire of behavior here represented as complex movement objects that relate to complex patterns of muscle movements in the environment. Now, how does this happen? Well, it is a complex story. Not only are parts of your cortex, the highest parts of your brain, involved in that, but non-motor areas involving emotional systems, the cerebellum, which we've talked about before, which is fine-grained control, rhythmic movements, patterning of movements in time, elements of your spinal cord, and then finally motor neurons that directly control the movement of muscles. This is a very complex system. Many people spend their whole lives studying it. And I have an example or an analogy, I think, that'll convey to you perhaps one way of thinking about how this whole thing works. Think about a piano. When you push the key of a piano, there is an amazingly complex set of elements, movements, physics that transforms the movement of that key to the sound that you hear. The physics of how that happens is amazingly complex, very subtle. But understanding the fact that you push a key and out comes a sound, and those keys can be varied, combined with other keys, etc., I think give a good analogy of how this movement object or vocabulary of movement would relate to the fact at a high level to the keys, and then ultimately which strings are hit by which hammers with which force as to movements of the muscles themselves. But there's a difference between the machinery we're seeing in your brain and spinal cord, shown on the right, and in a piano. And that difference is this system can learn. If you look at an experiment where a monkey sits at a desk, he is moving an articulated arm, and that articulated arm can have forces applied to it, so it's not a natural motor act from the monkey's point of view. and there's a set of numbered diagrams around the tablet that he's working on, the work surface. So he puts the pen in the middle with that arm. Let's say the light on number 4 comes on. The monkey's got to draw a line from the center out to number 4. Because there's an opposing force, he doesn't do it very well at first. The lines go all over the place. But as the monkey learns, it gets better and better. Now, this is being done unconsciously most of the time. The motor system, based on feedback, visually as well as proprioception, can get better and better at pursuing a goal, in this instance, drawing straight lines. It's like when you were young and you learned to color between the lines. Now, this is important because this is a form of learning, it's a form of memory. In general, it's different than the way we remember words or phrases or faces sometimes. But it is still a form of learning, and it's shared amongst all animals in terms of their brain. So this is a fundamental property of the motor system, is that it can learn. It can create new movement objects from old movement objects. So motor learning can proceed over time, and more complex behaviors can be learned. In general, like I say, this is unconscious learning. It's done with non-conscious parts of the brain. And in fact, the best way to disrupt the kind of recall of this learning is to consciously be aware of what you're doing. So if you're trying to work on your golf swing or your tennis return and you're having trouble, probably the worst thing to do is to start really paying attention to that, because you're mucking up the systems that are being in play here with the motor systems as they have learned an activity over time. Another interesting part of the motor part of your brain is sometimes it's smarter than the rest of your brain. What's on the screen here is something called the Ebbinghaus illusion. I hope everybody watching this would say that the orange dot in the middle of the large circles, as shown on the left, is smaller than the orange dot that's in the middle of the smaller circles, shown on the right. This is a classic illusion. I think since I'm calling it illusion, everybody knows that if you took out a ruler and you measured the size of those orange discs, they would be exactly the same size. But what if I built these discs out of wooden blocks, exactly the same size, and I put them in front of you, and I ask you to reach with your thumb and forefinger and pick up the disc in the center, either the left disc first and then the right disc second, or vice versa. If I measured the distance between your thumb and forefinger as you reached for these objects, I would find they're exactly the same size. So the motor planning part of your brain knows the state of the real world. The visual analysis part of your brain that is connected to the linguistic aspects that are making perceptual decisions for part of your brain doesn't know the real world. That sets up an interesting dynamic. Different parts of your brain could have different opinions, shall we say, about the state of the real world. And oftentimes that state depends on, are we analyzing them purely with sensory input, like visual analysis, and we get this illusion? Or are we analyzing the real world not just with sensory input, but with motor movement planning as well? And we could get a different result. Your brain has a more accurate picture at this time. And the point is there's a difference of opinion. And how those differences of opinion get resolved and what they might mean to future processing of the brain is going to be the subject of many interesting discussions we have in the future. Another item I want to talk about is the difference between moving your muscles and imagining moving your muscles. If you look at the top part of this diagram, this shows the areas of your brain that are active, and we know that from measuring blood flow, basically. When I would ask you to strum your fingers, so this would be a sequential finger movement, you'd be strumming your fingers on the desk, and we basically would see two areas of your brain active. Now in the bottom part of the diagram, this is the active part of your brain when I ask you to imagine moving your fingers. Don't move your fingers, just imagine you're strumming your fingers. Notice one of the same areas of the brain that is active when you're really moving your fingers, physically moving your fingers, is active when you're remembering. And then there's another part that is not there anymore. Now what this means, and this has significant implications, when you remember moving your fingers, when you imagine moving your fingers, some of the same areas of your brain are active as when you were actually doing the movement itself. This has implications about what really is involved in a memory. I mean, when we have a memory of something, when we imagine something, what are the components of that? And the main point I want to make here is even in the movement system itself, when you are imagining or remembering movements, some of the same neurons that are active when you're doing those movements are active even in mental rehearsal of motor actions. Not all movements are created equal in your brain. On the left-hand side of the slide, we see areas of your brain that are remarked at the top, physically related to parts of your body on the lower half of the diagram. This was found by stimulating parts of the brain and finding the muscles corresponding out on your body that would show activity when various parts of your motor cortex, or the motor planning or motor parts of your brain, were stimulated. What you'll see is that there is vastly more of your brain dedicated to your hands, your fingers, your face, especially your lips. And that means you have extra capabilities as it relates to the movement of those muscles and the movement of those limbs or parts of your body. Now what this means is this is not the exclusive control of those appendages, as we can see on the diagram on the right. Under the normal column, if you were to ask a monkey to grab a little piece of food in the well of a work surface, that monkey would show fine-grained control of its fingers as it grasped the piece of food out of that well and brought it to its mouth. It is using parts of the brain that are related to fingers and hands, etc., as shown on the diagram on the left. If you were to prevent those areas of the brain to communicate via the spinal cord to the rest of the monkey's body, the monkey would not be completely incapacitated to reach and grab. However, it would do so very, very clumsily. It would flop its hand over the well. It would have to, explore and grab around to get the little piece of food. It wouldn't be smooth. It wouldn't be targeted. It would not be precise. So I want you to see from this diagram that evolution has endowed our brain, and brains really of primates especially, very non-proportional representation of lips, face, and hands. In other animals, it might be different. It might be the whiskers of a rat or the cat. It might be other areas in a dog. The main point I want to get across here is that these areas of the brain show heightened sensitivity, a capacity for fine-grained control of fine-grained movements, but they don't exclusively control that. There are other areas of your brain that control gross levels of movement. And let's talk about a very, very important one next. The movement object we're talking about is locomotion. Remember, we're here because our ancestors were able to run away from the saber-toothed cat. And speaking of cats, it is in cats that we first found that locomotion is located as far away from the upper layers of the brain as possible. There's pattern generators in your spinal cord and lower levels of your brain that involve areas we haven't even talked about so far. So you can take the areas of the brain we've been talking about, the upper layers of the cortex, etc., and separate them from the spinal cord, and then take that cat and put it on a treadmill. This cat is basically walking on a treadmill under the control of only its spinal cord. As the treadmill speeds up, the cat goes through several different kinds of gaits, running, then basically galloping, and again, this is with no participation of the higher layers of the cortex. This is also true in rats. This is an example, perhaps a little easier to see, of a rat whose lower legs have been separated from his brain, so the spinal cord has been transected. He's in a very cute little walking jacket here, and he's on a treadmill. And those hind legs are moving completely independent of the higher levels of brain. This shows you that locomotion is a fundamental movement. It is a movement that is encapsulated deep within our nervous system because it's so important evolutionary to survival. So let's review where we are. We began this section noting that people appeared to have a movement template when they wrote this palindrome. It didn't matter what arm, it didn't matter what appendage was writing the overall sentence. It clearly was expressing an overall template of movement or a movement object. We then saw definitive experiments that showed there were neurons in your brain that represented a specific goal of the movement, obtaining a bit of food that was active regardless of which muscles were used to obtain that goal. So the movement object appears to relate to the goal, in this instance, get that bit of food. And it doesn't matter which muscles you have to move, the goal, the thing expressed by the neuron in your brain, is the object, the goal of that movement as the motor object. And then finally, we looked at more complex behaviors that were really objects of objects, so movements of movements, that represented high-order movements as these objects. And again, they're clearly related to some kind of goal, grasping an object, defending against a threat, bringing something to the mouth, chewing a bit of food. So I think the answer to this question is yes, there clearly are movement objects, and we can use our Homer analogy to explain this in a simple statement. So I think Homer would say, you plan with movement objects, you move with muscle stuff. Almost the exact mirror image of our sensory analysis. We said that you sense stuff and perceive objects, going from low-level analysis to high-order objects. The movement system, I believe, is the mirror opposite. Your brain is planning acts based on high-level movement objects, and they get translated by this very complex piano machinery into muscle stuff. Some muscles contract, some muscles expand, there's feedback, there's pattern generators for complex behaviors like locomotion, but there's a part of your brain that is planning on these high-order goals, these movement objects, that then gets translated to muscle stuff depending upon your position in space, what's in the environment around you, what you have to do to get the right part of your body to the right part of space to get the object, the goal of that movement, accomplished. Congratulations! That completes the first brain seminar on perceiving, sensing, and movement. The topic we'll talk about next is illustrated by this quote. The quote's from Amelia Earhart, a very famous American pilot, and what she said was, "The most difficult thing is the decision to act. The rest is merely tenacity." We've looked at how the brain can have different opinions within the same brain. Is the size of that orange circle the same or different? Well, how does the brain come to a decision about that? I mean, we studied the movement system, but the decision to act had already been made. We were talking about how do you implement this movement object. Well, it turns out that that's not as simple as you might think, and the subject of our next seminar is going to be the decision-making processes of the brain.